Hello and welcome to BIOS Frontier Science. I'm Kate Shannon, and today I'm thrilled to welcome Dr. Jeff Karp, a clinician, professor, and entrepreneur to the show. Thank you once again for joining us.
Hey, great to be here.
Awesome. So Jeff, to kick things off, could you please give us a quick introduction to yourself and what you're working on?
I'd be happy to. It's always a tough question to answer because it can come at it from so many different angles. First thing that jumps to my mind is I'm Canadian, born in a small town outside of Toronto, kind of farmland. So lots going on there that I could discuss. From like a career perspective, I'm a biomedical engineer. I run a research lab at the Brigham and Women's Hospital. My academic appointment goes through Harvard Medical School, and I have a number of affiliations in the Boston area. I'd say really there's two major goals in my laboratory, which is one is professional development. So everyone who joins my lab, I commit to being a mentor for life. And the second is translation. So trying to figure out how can we come up with ideas and make decisions in the lab to maximize the potential that what we work on can help patients as quickly as possible.
Awesome. And before we dive into the specifics of your work, like your background, it seems like there's many different angles to attack your projects. You have projects across a wide range of fields. Are there any common themes that tie your work together?
I would say, that probably the process that we approach problems. I would define, what we do as my lab is focused on the process of medical problem solving, and we're always, we're constantly evolving that process, iterating it, and applying it to new problems. I have deep curiosity about pretty much everything, and so I'm always interested to learn about new areas and unmet needs, and try and figure out, really from like an engineering perspective, what's that really small fragment of information that is most critical to focus one's attention on, to define the problem in a way that others maybe have missed certain insights. And to me, so it's really about like applying problem solving skills and focusing on problem definition and the process of how we advance our experimentation towards uncovering new insights. And so that's one theme that I think is in every project. And most projects, not all, involve some aspect of delivery, whether it be delivery of small molecules to the body, delivery of materials, or delivery of cells. But there's a whole bunch of other projects that we work on that don't have a delivery component, but I would say the majority do.
Interesting. And so jumping into one of your earlier projects around stem cells, so your lab was investigating the use of small molecules to activate stem cells. Could you give us a quick overview of this?
Sure. I think the area of cell therapy is incredibly exciting, and that excitement just builds and builds pretty much every year, every day. And I think that one of the main modes of cell therapy is to take cells out of the body, manipulate them. Sometimes that involves culturing them in certain ways or expanding cell numbers and then putting them back into the body. And that's an incredibly complicated process that has many quality control measures, because you have to make sure that at each stage in the process, you kind of have a handle on what you have. And because it's a living product, cells actually will change depending on their environment. So if you have small changes in how you're growing the cells, the cells themselves will change a lot. And so there's a lot of complexity there. At the same time, there's incredible potential. And we've been able to realize some of that potential as a field recently with cancer therapies, CAR T cells, for example, and there's been a number of other examples. But a number of years ago, we started thinking, is there a way to simplify this process? And that's also a common theme, I think, in a lot of projects in the lab is trying to approach things from the perspective of radical simplicity. And actually going back to one of your previous questions, all these things kind of jump up like after the fact, but it's a lot of projects involve looking to nature for inspiration. And so, we can get into that if that's relevant later. But so we started thinking, well, instead of taking stem cells out of the body and manipulating them and putting them back in the body, could we do this by, if we could understand the location of stem cells or progenitor cells in the body, and we could understand how those cells interact within their environments, could we deliver small molecules, which are kind of like standard drugs, to the stem cells and be able to manipulate them in the body without having to remove them? And that began a significant effort in the laboratory with a number of collaborators to try to identify the relevant signals, to try to figure out what's the best first application to focus on. We started in the intestine, and we moved into the cochlea with a focus on hearing loss.
So actually, I'd love to hear more about that. I mean, this is a fascinating approach with, I think, a lot of potential applications. But when you spun out this technology into frequency therapeutics, you chose hearing loss as your first indication. Why was that?
So initially, we were focused entirely on the intestine. We actually started off with a project that was focused on trying to tissue engineer intestine. There's various conditions. There's one called short bowel syndrome, and we were trying to develop longer segments of gut tissue that could be transplanted, or could we do this actually in the body? And that ended up being incredibly complicated, and the biology just wasn't there for us to do much. And so we focused on the lining of our intestines, which is arguably one of the most regenerative tissues in the human body. It regenerates every 4 to 5 days, completely turns over. And this is governed by a very small number of cells that exist in the base of crypts. So in your intestine, you kind of have these valleys and you got these peaks, that kind of go like this to expand the surface area for absorption of nutrients. And at the very base of the valleys, you have these stem cells. And those stem cells are constantly dividing throughout life. Every 24 hours or so, they divide, and then they keep proliferating, and they become all of the cell types. Essentially, they differentiate is what we say, and they become all the cell types of the lining of the intestine. And so what we did is, through understanding the cues in the lining of the small intestine, we found small molecules that could manipulate these stem cells to control them in ways that no one had ever done before. And that led to all kinds of different projects. And when we approach problems in the lab, I'm constantly kind of diving in, and then, I jump out and say, okay, was there anything we could be working on that is higher impact? Or, because there's a tendency, I think, in academia, maybe in life, to just trying to get into this incremental mode where you have, something really interesting happen, but then you just kind of push it along in these baby steps. So we stepped back and we said, well, what could we do that would really be transformational? And we were just looking around trying to think about it. And somebody who I was working with, Xiaolei Yin, said he found a paper that showed that the cells that make hair cells in the inner ear had very similar properties to the cells in the intestine, these stem cells. There's a receptor on the surface called LGR5, and actually both the intestinal stem cells and the progenitor cells in the cochlea, the shell-shaped structure in the inner ear, had that as well. But the big difference was that in the intestine, those cells are dividing, as I mentioned, every 24 hours. But in the inner ear, they don't divide after birth. They just remain dormant for life. And it was discovered that those cells can make new hair cells. Now, and I know I'm kind of providing maybe a lot of detail here, but I think it's important just for context. We're only born with 15,000 hair cells per inner ear, and they never ever regenerate. And they are the cells that respond to sound waves and then the cell connects to a neuron, and that goes to the brain. So that's how we're able to hear. It's just by those really small motions of the, and it's not actual hair. It's called stereocilia, but it looks like hair. And when those cells die from exposure to loud noises or even just the aging process, and there are certain types of drugs, they never ever grow back. So you start with 15,000, and over time, those numbers just decrease and decrease. So your hearing just constantly gets worse throughout life. And so we started thinking, well, if the cells that are responsible for regenerating the lining of the intestine have similar properties to the cells in the inner ear, which can make new hair cells, but they never divide, could the molecules that we discovered that could control the lining of the intestine work in the inner ear? And we tried it, and it worked. And so that led to a whole series of experiments and collaborations, and then eventually the launch of Frequency Therapeutics that you mentioned.
That's awesome. And so you've talked about hearing loss and, in the intestines, but what other disease indications or applications do you think this technology could be applied to?
I think this could be applied to many different applications, many different problems. Anywhere where we have knowledge of the signaling events, how cells are communicating in the body, in and around stem cells or progenitor cells, this type of approach should work. So could be, it's a whole range of things where this could be useful. You could think of regenerating smell, for example, perhaps regenerating eyesight, kind of think of the senses and then beyond.
Awesome. And so what comes next? What are some of the future milestones and next steps for this technology?
So currently, the company Frequency Therapeutics is examining these molecules. It's actually a combination of molecules, and they have a whole host of different combinations that they've discovered. And so examining this in clinical trials, and that can be a very lengthy process. No one has ever done this before, developed a regenerative therapy for hearing loss. And there's so many different segments of the population, those with sudden hearing loss, maybe it was from, some explosion, or could have been someone was in a band, for example, and an instrument went off. Or could be just from the aging process, or even drugs, certain antibiotics or chemotherapeutics can cause hearing loss. There's so many different segments of the population where hair cells can be damaged. And the company is now advancing this, in a number of different populations through clinical trials. And I think, there's a lot to learn because no one's ever done this before, trying to figure out the dosing. But the early signals look promising. And so the company's moving, full speed ahead, trying to, it's one of these things where you're trying to learn as much as you can, and then kind of repoint, where you're kind of firing your next, arrows to see if you can, make more and more progress.
Awesome. So Frequency is clearly doing something that no one has really done before. What do you think are some of the biggest challenges for them moving forward?
I think, one of the biggest challenges, and I think this is actually common for, any type of new therapeutic, is that, a certain amount of biology that we know, but there's also a lot of biology that we don't know. And I think that, the challenges really tend to be around, discovering what you don't know as you go along. And so, the team, they have a clinical advisory board with, many different people who are looking at this from different angles, scientific advisory board, again, lots of different angles kind of looking at this. And as new information comes in, it's trying to assess how best to interpret that data, and make the best decisions possible to move forward. But again, when you're developing something new, there's always unknowns. And I think that, that to me is the biggest challenge is how do you conduct experiments so that you uncover the insights that you need to learn to then have a better chance of getting closer to the target on the next experiment.
Oh, I think that's very true. And so, moving on a little bit, your lab also has some other stem cell projects that you guys are working on. Some of it, really on the cutting edge. One of them involves controlling cells with intracellular particles. Could you give us an introduction to this?
Sure. So this is actually one of the first projects that I started when I formed my lab several years ago. And it was really kind of coming back to the traditional cell therapy where you're taking cells out of the body, manipulating them and putting them back in. And, I often look at things through an engineering lens because my basic training was in chemical engineering. And so just kind of reflecting on as the field was evolving, I realized that when we put cells back into the body, and often the results are kind of all over the place. I was thinking like, okay, well, those cells are at the mercy of wherever they are delivered to. So, if you inject them into the heart versus into fat tissue versus into muscle, for example, they're going to behave completely differently because they're living therapeutics and they respond to their environment and change, And so I started thinking, well, okay, well, how can we better control cells following transplantation? Because, this could really make a huge difference if we're able to deliver cells, and irrespective of what environment they end up in, or how, as tissues age, the different signaling can change as well. And so you don't want to have a therapeutic that is just changing in every single patient that you inject the cells into. You want to have as much confidence and predictability as possible. And so what we did is we said, okay, well, lots of groups were putting cells on top of materials and then transplanting them, but you can't inject those cells into the bloodstream, because it would cause a stroke. Many places where you couldn't deliver that. And so we said, well, what if we turn the problem upside down? What if we flip it on its head, and instead of putting cells on top of materials, what if we put materials inside of cells? And so with this concept of radical simplicity in mind, what we did was we came up with this concept that we would take small particles, just a micron or so in size. Human hair tends to be 100 or 200 microns thick. So this is, 100 or 200 times smaller than a human hair. We take that particle and we would make it out of a degradable material, something that would degrade in, a few weeks, a month or two type of thing, like degradable sutures, for example. So we took that material and we put into there just very simple molecules that were already, for example, in approved drugs. And what we did is then we fed those particles with the molecules inside of them to cells, and the cells internalized those particles. And we found a certain formulation where the particle remained in the cell. Sometimes the cell will spit the particle back out because it doesn't like it there. But we found compositions where it would be stable. And then what happens is these particles slowly degrade over time and release the molecule, which can then control the cell behavior. And so it's a way of controlling the cells from the inside out following transplantation. And so this has led to a whole range of different sort of trajectories for that project. And then a company has licensed that technology and is now looking at, moving that to patients.
Very interesting. And so where do you think this technology can be applied, and what do you think the most promising initial applications are?
I think there's a lot of possibilities. I think anywhere where you have cell-based therapy that is being delivered to patients or being proposed to deliver to patients, I think it would be applicable because we can better control, for example, what the cells secrete. So if you're using the cells for regenerating muscle, we can program those particles to help the cells produce more muscle tissue, for example. Or if we want to regenerate another tissue, we can program that. A lot of cells tend to die following transplantation just because of the shock of going through needles and then ending up in tissues. And there's often, blood that forms, and then there's a blood clot, and there's not a lot of movement of nutrients in blood clots. And so cells can die. So we can deliver molecules that can, help reduce the stress in cells to be able to prolong their survival so that they can actually function properly.
Very cool. All right, so moving on to kind of another area that your lab is focusing on, tissue adhesives. You've developed several products in this space. Could you give us a quick overview of these projects?
Sure. So we've been working on tissue adhesives. Actually, I started working on this when I was a postdoc in Bob Langer's lab at MIT. We developed a gecko-inspired adhesive. Actually, I was walking around the lab one night. Most people had left, and I was kind of looking around, minding my own business, kind of just thinking. And I noticed a journal article that was open with Spider-Man, and I was like, "Hey, what's that?" And the authors had learned how geckos can walk up walls and hang from a single toe, and there's no glue, there's no suction cups. It's just these tiny features on the surface, and they're hierarchical. So they kind of go from like a small feature, lots and lots of them, to then like a larger feature and a larger feature. They're all attached, and that maximizes how those geckos can attach to surfaces. And it just spreads out really nicely onto surfaces and makes things called van der Waals forces, which most people probably learn in college but forget about them because they don't really appear to be that important. They actually one of the few kind of places where they are important, because you just get so much surface contact. And so what they had done, this group, they had studied geckos and then made a dry adhesive tape. And their example was they had stuck a Spider-Man action figure to the ceiling. And this was like in a Nature journal. So I was like, wow, that's really interesting. And that got me into the whole world of bioinspiration. That's how I started looking at nature for inspiration. And I never really thought about it before, although I'd kind of grown up in farm country. So I had that incredible experience, which shapes actually a lot of the ways I think I look at things today. But that kind of set me out on this whole journey because we had been making materials and looking for applications. So we initially developed a tape that was unlike the ones that others had developed before that were for moving glass sheets around in factories and things like that. We developed a biodegradable, biocompatible tape-based adhesive that could be placed almost anywhere in the body based on this gecko type mechanism of adhesion. And as we started working on that, clinicians started, all sorts of doctors are contacting us and asking, telling us about all kinds of problems. Like there's, resection of lung that often you get leaks and there's no way of solving that, or various surgeries, like types of urology type surgeries, or holes are left behind. They're really difficult to seal, and nerve reconstruction. We just kind of heard about all these different challenges. And so that, really helped us build momentum towards developing new types of adhesives. So we went on to develop all kinds of different tape-based adhesives. Many were inspired by various creatures in nature, porcupine quills and parasitic worms. We developed a glue that was inspired by various sea creatures, slugs and snails, like sandcastle worms, for example. And so we had maybe 5 or 6 different adhesive technologies, all designed to work inside the body, which is extremely challenging because most adhesives, if you get them wet, they just stop working. And so we had to think about this completely differently than most of the adhesive technologies that were available. And then when you put something in the body, you really want it to degrade over time. And so there were all these constraints that we were faced with, which I always find exciting, because when you constrain a problem, it actually can help you solve it. It also makes it more difficult to solve it, but so it's like this interesting balance. But anyhow, we were able to develop this surgical glue, and then we formed a company called Tissium, which is based in Paris. They were able to get regulatory approval for that, for vascular reconstruction in Europe, and now we're moving towards starting a trial in the United States.
Interesting. So, I mean, you kind of outlined some of the challenges around medical adhesives, but, what are the biggest challenges there? And then, what are the applications that you're kind of pushing some of these adhesives for? And, what are doctors using them for?
Yeah, well, one of the biggest challenges that we faced early on is manufacturing, because something that as an academic, I used to never think about. Now I think about it all the time. because we were making this material, we made it in the lab, and it took us lots and lots of different iterations to find materials that would actually adhere to wet tissues in these studies we were doing. But then we were making those materials like 1 gram at a time, 10 grams at a time, just, it's very small amounts, like tablespoons kind of thing. And we were using them right away. So when we started the company, they were like, okay, well, we need to make kilograms, and this has to be shelf stable for a year or maybe even 2 years. And so we started to do that, and it just didn't work. We weren't able to manufacture it. And so we had to kind of change the formulation, and it took a lot of experimentation. We had to bring in a bunch of consultants, chemists, just to help us think differently about this. And eventually we were able to figure it out. But that was an enormous challenge, just to figure out how to manufacture this and have it be shelf stable. Often these materials have some degree of reactivity to it, and so you don't want it to react before it contacts the tissue. You want to have repeatable, predictable, function. And so that took a while to figure out. The other thing I would say, which is quite challenging, and I never really appreciated this before, which is when you have a technology that's a platform that can be used for multiple applications, it's often very, very challenging to figure out the first application to focus on, where, and that involves, I never thought that it would take so long and so many minds coming together to figure that out, but it always does. And the reason I think is because there's, technology risks, like different applications. If you're putting something inside the heart, for example, it's a very high regulatory bar, and there's a lot more, experimentation you need to do versus if you put something on the skin. But then there are all kinds of adhesives already that go on the skin, but there's very few that would go in the heart. And so, it's like, what's the market opportunity? What's the technology risk? What is the regulatory path, the risk of regulatory and the cost, the costs are very different to go after different applications, the ability to put teams together, ability to recruit patients, like it's different for all of these things. So just figuring that out. And then also it's tough to figure out, when you're having conversations with companies and doctors, everyone kind of has different perspectives. And so you have to have a process, a way of thinking about what is truly the best application and kind of rank ordering them, where everyone you talk to says this application is the best, or something different, or it's really difficult to figure out. So to me, that's kind of one of the biggest challenges. Tissium, they're advancing now towards a number of applications. One is vascular reconstruction, another is nerve reconstruction, and then another is hernia repair.
Interesting. And actually, I want to quickly go back to that, because I think that is a challenge that most biotechs face, is picking that first application. And like you mentioned, there's so many trade-offs that you have to consider. How did you go about picking those first applications, or do you have a framework that you use to help you decide?
Yeah, the framework, if I was to try to boil down what the framework was, and it's I would say, highly iterative process, engaging many, probably dozens of experts, meaning doctors, people in industry, others who have developed similar products before, and just asking questions, taking lots of notes, going over it, trying to ask better and better, more focused questions, as you go, picking, one or two kind of leads, advancing them, and just constantly keeping your ear to the ground, having discussions about the reception that you get as you're making progress. And sometimes you need to change. Sometimes you're headed in one direction, so you have to make a decision at some point, because perfect is the enemy of good, as they say, and you need to advance and gain momentum. But I think you also have to be open to reconsidering, as you go, and then big part of that is having enough resources to then fund that. And so you need to want to make sure that when you're raising money, that you have enough funds so you can take a few shots on goal and not just one.
Makes sense. And so you mentioned some of your first shots on goal that you're taking with these CCA adhesives, but what are some of the other potential applications, either within healthcare or outside healthcare, that you think are or might be interesting to pursue?
I think one really interesting application in healthcare is to use new tissue adhesives for minimally invasive procedures. And so these are procedures where you have very small incisions in the body. Often it's difficult to tie knots, sutures in small spaces. So if you can't do that, then you can't perform the procedure. And so I think that when you have adhesives, which I think Tissium now has, that can work inside the body, even in the presence of water or blood, for example. You're now able to perform minimally invasive procedures you weren't able to perform before. And often when you do things like that, patients heal faster. You're able to have the scars are much smaller. And you're able to do things that you can't do right now. And so to me, that could just by having improved tissue adhesives, that could open up the entire minimally invasive surgery space. I think there's lots of applications, but that would be one that I think would just be transformative.
Awesome. And so switching tracks a little bit, you've obviously developed a wide range of products that you've been able to successfully spin out into a couple of different companies. I wanted to ask you some questions about that. The first one is, how do you go from an idea, a technology, or a discovery to a useful project or product that has an actual application?
Yeah, so one of the things that I would say, okay, so I think one of the best decisions I ever made in my career was coming out of my postdoc and going into my faculty position and realizing that I had some great mentors like Bob Langer and others in my PhD um, that had started companies, and I could kind of see what was possible. But I think doing it yourself is very different. And I realized, really try to develop a keen sense of self-awareness and kind of clue into, the things that I don't know, of which there are, most things I don't know. So what I realized was that I need to, and I was really, in addition to mentoring, and I spent a lot of time kind of studying mentors and trying to figure out how to do it in the best way possible. I realized that I really wanted to translate technologies, but I didn't have the skills to do it. And so what I did was I made the decision every 2 to 3 weeks coming out of my postdoc to meet with somebody within the entrepreneurial ecosystem, be it a patent lawyer, a corporate lawyer, reimbursement, regulatory expert, manufacturing expert, entrepreneurs, people in industry, other, entrepreneurial faculty, for example. And the goal wasn't just to meet and network. The goal was to form relationships with people. And so, over time, what happened was I was able to keep people up to date on various projects and start to ask for advice. And so what ended up happening is that served in so many different ways. One, have a lot of friends now who it's always kind of like a community, which is I think really great to kind of you always need as much support as you can, and so that's been really great. Another aspect of it is to create filters. So as we're advancing in projects, I've realized that there's risks that we're constantly evaluating and making decisions based on those risks, and by having people that I can reach out to and say, hey, do you think this could be manufactured? Or, do you think we could patent this? What's kind of the landscape in this area right now, trying to figure out which application to focus our experiments on. So I'm constantly it's almost like an informal advisory board that I'm interacting with and they're helping me to make decisions. And then as things go along, those people, a lot of them are interested to be involved, if things are going well, in terms of, helping to start a company or join a company that may form. And so that really has, been a big big part of it. And also, another part is always, trying to evaluate when is the right time to start a company. I think you always want to keep things in the academic setting as long as possible, because there's often so much de-risking that you want to do. Once you launch something and you're able to get some investor dollars into the company, the clock is really ticking, and you have to make really like the decisions that you're making are just really high pressured, and there's certain timelines money's going to run out. It's really scary, actually. It's like, you look at these curves and it's like, all right, we're going to run out of money in like 4 months, and like, oh my god, like, so whereas in the academic setting, we can apply for more grants, we can be more exploratory, there's not as much pressure on. And I found that one of the greatest risks in everything that we do is the biology risk. So if we advance things too early, what can happen often is that there's certain aspects of the biology we don't understand, and that can eventually just, everything can implode because you just don't know where to go because you don't have enough knowledge of the biology. And so we want to make sure that we, as we're advancing in the lab, that we understand enough of the critical biology. Either others have made the discoveries or we've kind of explored. And so that's kind of the process. So it's a matter of kind of iterating, having this deep network of kind of informal advisors, friends, I'm constantly interacting with. And then as we're gaining momentum, people in the network, either them or they know people, will then join the effort, we'll start the company together. And then what will happen is that will gain a lot more momentum because, the entrepreneurs, for example, have all kinds of people that, they work with, who want to work with them again. And so, then that starts the whole process, and they have investors that they've worked with before. And so, that can really lead to a lot more progress and momentum.
Oh, that's awesome. And it's great to hear that you put so much effort into this and are developing the skill. And I think the results clearly show, both in the success you've had spinning on companies and even the way you're just able to talk about this stuff. But how do you choose what to spin out, and what motivates you to spin out a new company?
Yeah, I think so. Two very different questions. I think one is, how do we choose what to spin out? I think that is really just about having lots of conversations, and sort of pressure testing things, putting things together. Maybe I'll reach out to some investor friends of mine and say, okay, what do you think about this? And what would be your main questions? And then maybe we go back to the lab and try to address some of those questions. And once it kind of feels like, we've answered some of the most critical questions, because I think a big part of it is you want to de-risk it enough. You also, I think, want to have at least one, if not many, peer-reviewed publications, so that this has gone out to the scientific community in some format, usually a manuscript. Others have evaluated it, have asked, tough questions, gone back, addressed those questions, and then have been able to publish that work. So I think that, to me, is how we decide. We want to have ideally a strong patent that can block others, not just block others for like the narrow composition that we've developed, but ideally block others from, doing the same thing with a different material, or, finding ways around the patent. So that often can take a lot of time as well. And then your other question was about, so maybe go back to if you could say that again, that'd be great.
Yeah. So what motivates you to spin out a company?
Right. Yeah. And then for the motivation, I think that there's a number of things. What I've realized is early on, I'm committed to wanting to, help patients as quickly as possible. And I realized that the best way that I've seen others do that is to start companies. And I think the reason why that ends up being the best method is because when you start a company, you're a co-founder, you're involved in a lot of key decisions that are being made. Is like, you never want that to die, because you've worked on it for so long. You invested so much of your time, and you've sacrificed, your time away from other things like family and things like that. And so I think that there's always challenges, I think, in transferring the technology from the academic setting into a business, but then also so many things come up along the way, where it's relationships with consultants or other people, advisors, people in academia who could help. I mean, you always need many more people than you think you're going to need. And to me, it just, starting companies, it just seems like, from my experience, the best way to move technology forward, remain involved, and really help to generate the momentum. And I think, you can also generate patents and then license the patents to companies, but often, and we've done some of that, but often you tend to be more arm's length from the development of that technology. And in companies, they always have like reorgs that are go on, and people, the champion, the person who really wanted to bring it forward leaves, or they make a strategic decision in the company to no longer work on it, or to kind of narrow the fields that they're working on, and so things can easily die. Whereas if you start your own company and you're involved, there's less chance of that happening.
Interesting. So you've touched on this last question at a couple different points, but what are some of the key attributes you think are necessary for a successful spin out of a new company?
I think there are many things. I think that there may be like a number of, let's say, like obvious things, but then I think a number of less obvious things, and maybe focus on some of the less obvious things, at least less obvious to me when I started. So things like the ability of the executive team or the operations team to realize that there's constantly going to be holes in their vision. There's constantly going to be things that they don't know. There's going to be assumptions that have been made that are not good assumptions. And to find like the most critical minds around to find those holes, and constantly be helping to identify what those holes are and try to fill those holes, or to help figure out what are the best experiments to perform to generate the insights that we can make better assumptions. And to me, so it's that kind of, trait of the people who are running the company that I feel is really, really important. And often, companies where they start, where they initially kind of point their North Star, it changes over time. And so the companies, you need to have people in the companies who are able to evolve, and sometimes they'll evolve very quickly, and just be open to criticism and be able to, work together and I think, another big part, actually another thing I've noticed is teams that have minimal ego, because often, if you talk to CEOs, and I've talked to many, they will say over time, as companies grow, they spend most of their time managing the people and not focusing on the vision or the execution of the plan. They're just managing the different personalities, in the business. And I think that, to me, that's another big thing is you want to create as lateral an environment as possible. Sometimes you need hierarchy, to get things done and to move forward. But I think that's another key thing that I've noticed is teams where people have their ego in check, and they have a ego management plan, in their minds, ways of managing, and sort of that. I feel like those are the teams that end up executing the best and lead to the most successful outcomes.
I think that's some great insight. So we're getting close to the end here, but before we close out, I wanted to ask you a couple last questions. First is, where can people learn more about your work or the companies that you've helped found?
Yeah, so we have a lab website. It's very original, karplab.net. I had this name for my lab that I had created, and I thought everyone was going to use it, but people just started using Karp Lab. I guess it was like just too complicated. So in the spirit of simplicity, we've just stuck with Karp Lab. So karplab.net. And then there's, we're publishing our work. So if people are, scientifically inclined or interested to kind of look at publications, you can go to PubMed and search for our papers, or they can contact me. I'm always happy to send people papers, or go to some of the company websites of the companies that we've spun out of the lab. That's usually a good place too.
Great. And do you have any shameless plugs or last thoughts?
Shameless plugs, I guess. Geez. Well, I'm writing a book that will be coming out next summer. It's called Lit, L-I-T, and it's going to be published by HarperCollins and translated into a number of languages. And it's essentially the process that we've been applying in the lab and applying it to much broader sort of things, like just how to advance in your life with a process, with a strategy that's really focused on purpose. And I have realized, over time, that I initially was really focused on productivity, and I have made the transition to focus on purpose as a main goal. Clearly, being productive is important, but making that switch from productivity to purpose is really important. And the book is a lot about that, and how we can really think deeply about how we exist within the ecosystem and make decisions that are really best for the planet and best for ourselves. So that's one of the things. We also started, actually, just to name one of the things, we started this company called One Fun Company during COVID to help children change their habits, their hand washing habits and other personal hygiene habits. And we invented a new soap formulation called Splats, which are it's kind of these tactile gel balls, and kids grab them and they squeeze it, and the soap kind of splats out of it. We've done all kinds of surveys, and it's actually really changing kids' hand washing habits, which we think has been quite important during the pandemic, but also coming out of the pandemic. And we're in the process of developing other products as well. And then a portion of the proceeds go to children's mental health efforts. And so, yeah, that's something else that we've been working on in the past year or so.
Oh, that's awesome. Two great initiatives, I think. All right, Jeff. Well, thank you so much for an absolutely incredible episode. This was a lot of fun, and we're really grateful for your time. This was awesome.
Yeah, no, it was great to speak with you. Thank you.
All right.